And the next speaker we have is Dusha, and Dusha is a molecular biologist, and she's also part of the Berlin Soapbox Science team. And she will be talking to us about genome engineering and actually about the CRISPR revolution. So I'm sure questions you had may still apply.
Yes.
Okay, Dusha, the floor is yours.
Hello, everybody. Thanks for joining. I'm Dubravka Vuc is my full name. I'm a postdoc at PIMSB MDC here in Berlin. And today I will tell you about this new revolutionary technology that was just awarded a Nobel Prize, and to two female scientists, Emmanuelle Charpentier and Jennifer Doudna, for the development of a method for genome editing. And just like many other groundbreaking discoveries, this one came from a very basic research project. What they studied is how do bacteria fight off a viral infection. These are little viruses, and this is the bacteria. And bacteria have developed their own immune system to fight off viruses, and this is how it works. When the virus invades bacteria, bacteria stores a little piece of the genetic information of the virus within its own DNA in the form of this CRISPR array. This information is then copied into a dynamic form of genetic information, RNA. In this case, it's called guide RNA. This guide RNA can then team up with a Cas9 protein, so-called molecular scissors in the cell, and they too go around and look for invading bacteria, where the guide RNA serves almost like a GPS system for these molecular scissors. And once there's a match between this guide RNA and the invading DNA of the virus, the Cas9 protein cuts the DNA and disables the virus, and in this way the bacteria survives. And this is a nice evolutionary story, how do bacteria fight viruses, but what scientists very quickly figured out is that since most living organisms store their genetic information in the form of DNA, with this tool, we are actually able to, by just providing the guide RNA, make cuts in the DNA of any living organisms, microorganisms, plants, animals, and even human DNA. And this is how this looks like. And this is very important because every single cell of our body has exactly the same genetic information, exactly the same DNA. And what differs between different cells is which portions of the genome are active or being copied into RNA, which can then make a protein or act on its own. So with this tool, we can cut the DNA and examine its effects. And although one of the greatest achievements of the previous century was that we have sequenced the human genome, the DNA, we know only about less than 3% of its function. And with this tool, we can learn about the human genome and other genomes. And in addition to cutting, we can also do all sorts of different things with the CRISPR-Cas9. We can, for example, label places in the genome or make very precise edits of single letters, or even switch genetic elements on or off without even altering the sequence at all. In addition to this tool being very, very precise, it's also scalable. So we can assay tens of thousands of genetic elements in the form of so-called CRISPR screens. In a single experiment, in a single dish, we can filter out which of these genetic elements contribute to, for example, cellular growth or resistance to a particular drug treatment that we provide, and so on. And this is, for example, what I do in the lab. I use CRISPR screens to examine the regulatory landscape in neuroblastoma, which is a devastating childhood cancer. And like we heard from Liliane, we could use CRISPR to edit stem cells and differentiate them into neurons, for example, or cardiomyocytes, like she showed, and study them in a cell culture dish, or even use them for therapy. In addition to working with cells, scientists all over the world have made different disease models so that we can study the disease on the whole organism, or just the effects of the CRISPR system on the whole organism. A lot of work has been done in agriculture so that we can make, for example, crops that are resistant to diseases, and therefore we wouldn't have to use pesticides, or people are working on making multi or super nutritious vegetables or gluten-free crops and so on. And just like bacteria fight off viruses, we can also use this system potentially to either treat or detect viruses. So now in this current pandemic, one of these fast detecting kits for COVID-19 is based on the CRISPR-Cas9 system, and there's even research that shows that potentially we could use this as a therapeutic for COVID patients. And we could also use it to treat diseases. A lot of diseases are very complex and we don't know their genetic basis. A lot of things go wrong. But for a certain number of diseases, only one gene is affected, and even only single letter within the DNA is messed up, basically. And with CRISPR, we have the tool to change this and cure patients. And there's several ongoing clinical trials of which, and we have already a few cured patients too that had beta thalassemia and one with sickle cell anemia. Also, we can try to use the system to cure more complex diseases, such as, for example, cancer. We know that cancer survives in our body because it escapes our immune system. So what some clinical trials are trying to do is to isolate the immune cells of the patient, then edit them so that they could recognize the cancer and then put them back into the patient with the hope that they will eat up the cancer. And I think we all agree that this is a wonderful tool for curing different diseases, but with this technology, we also have certain ethical concerns, because not only can we edit somatic cells, so the cells of an adult organism, but we can also make changes in the embryos, and in this way, we can change the genetic makeup of humans forever, because these changes are then inherited. So to make sure that this tool is used in an ethical and safe manner, scientists are working together with governments and ethicists to really make sure that we are doing the right thing. But not just scientists, I think we all need to think about and learn about this technology and make sure to know where do we draw the line between therapy and enhancement, because this line can be very blurry. So we need to think about that once we get the knowledge tool, what is it that we want to cure? Do we want to make humans with a perfect eyesight, or with the ability to eat high-fat foods, or have big muscles? Or once we get the knowledge tool, do we, for example, want to cure aging? And who would be the ones who get these privileges? And we should wonder also, is this human engineered superhumans, are they the next logical evolutionary step for us or not? And I would like to leave you with those questions. Summarize, I hope I convince you that CRISPR is an amazing tool for molecular biology, enabling us to do much more research. It has the potential to fight off and cure diseases, but we need to make sure that this tool is used in a cautious manner and that it has a positive outcome for both us and for the planet. And thanks to BIMSB, Soapbox Science, and Teachsurfing, I encourage everyone to join Teachsurfing. It's an amazing platform. I'm happy to take questions.
Thank you, Duza. Thank you so much also for this reality check. Maybe whilst we wait for some questions, you could tell us a little bit about something that Liliana also mentioned. What should we expect in terms of the timeline of these things? You know, they're already being used for some things, but some things are still being developed. We should be careful that they're not ready to be used or not. So what kind of timeline are we looking at? What should we expect?
Well, it really depends on which on the disease. Yeah. So this, as I said, for most diseases, we really don't know what the cause is. For some of the monogenetic diseases like beta thalassemia and sickle cell anemia, we already have clinical trials that are successfully treating these patients. There's also easier things to edit than others. So if we can isolate the cells and edit them in a cell culture dish, that's one thing. But if we have to treat, we cannot reach all the organs, or we don't really know how safe it is. So all we can do is for now just edit things outside of the body and then put it back without, as somebody mentioned, there might be an immune response to the Cas9 system, to the guide RNA. So this is all something that's, it's ongoing research and we still don't know.
Maybe can you tell us a little bit about, this is a really complex line you were talking about that we need to navigate, right? Where do we draw the line between curing and enhancement? So what other fields of science are focusing on this? Are there like social scientists or lawyers, or like who is focusing on this line, and how can we learn how to better think about it?
That's a great question. A lot of ethicists are involved in this issue because this line is really depends on the culture. Yeah, it's like if you are, for example, curing yourself from a particular disease, and then why don't you just add something to make you have a perfect eyesight, for example. So it's hard to, once you start doing it, we saw this even from plastic surgery. Yeah, once it just goes on very rapidly, exponentially. So a lot of people are involved, both from social sciences, governments are involved, because we need to put rules in place that are going to be applied everywhere. So not that we have one set of rules in Europe and then they can do whatever they want in China and Russia or so on. So this is a bit of a political game also.
Isn't it always?
Yes.
A question from Liliana for you. So can the CRISPR system be used in every animal or only in humans and bacteria?
Yes, so most organisms store their genetic information in the form of DNA. So we can edit plants, animals, microorganisms. We are actually, I didn't mention it, but we also have the tools to edit RNA. So we can even target RNA viruses such as the SARS-CoV-2.
Which brings me to Floor's question. Do you think that CRISPR could be used to mitigate the impacts of COVID in severely sick people, like to treat the lingering effects and associated diseases?
Lingering effects, that's a pretty big definition, but there was a paper in Cell that showed that by using 6 different guide RNAs, you could theoretically treat these patients. So basically use the CRISPR-Cas9 system, deliver it to the lungs of the patients, and they would then cut out the virus. Yeah, but of course, this is far from being applied because we are not, as I said, not really sure how this will affect if we give it directly to. But it's widely used in this CRISPR detection tests. Sorry, COVID detection tests.
Okay, well, thank you very much for answering all these questions. We will soon